Hi, my name is Paint, number 11, and today I'd like to present about my summary notes. So let's start off from chapter 1. So first is about the science of life. How can we know that is it the living things or non-living things? So we can know from this one. 7 characteristics of all living organisms. First is the cellular organizations, which means that all living organisms need to have cells. Second is the ordered complexity. Third is the sensitivity, which is the reasons that we are respond to some things or we react to something. For example, I touched the hot surface and I jumped or I pulled my hand out. That is my reaction because I respond like that. Fourth is the growth, development, and reproductions. Now, reproduction is the key. I think it's the main key of like the characteristic of living organisms. Because like if we can't reproduce, we're not living things at all. 5 is the energy utilizations. 6 is the homeostasis, which in Thai we call it the การรักษาสมดุล. 7 is evolutionary adaptations. Okay, so next we're gonna know about how living things organize, like what is the systems of living things. So it's this one, the hierarchical organizations. So the first level is the cellular level, which is those atoms, molecules, macromolecules, organelles, and cells, which is the smallest levels of all levels. Next one is the organismal level, which is bigger than the cellular level, because the organismal level is when like a group of cellular level together, like cells when they form together, they form a tissue, something like that. Then we have tissues, then we have organs, which is bigger than tissues, and bigger than organs we have systems, and then we have organism, which is the living things, like the individual living things. Next is the population level. Population level is when the group of living things are together, because organism is the living thing, right? Is the like individual of the living things. Okay, so the populations level is the populations, which is when one species of living things are living together. Next is the community. Community is when 2 or more species of living things are living together. Next is the ecosystems level. It is when all living things and non-living things are living together. Like in the forest, like that. We have living things as those animals, like tigers, snakes, like that, or frogs, like that. And we also have the trees and grass, like that, which is the non-living things. And then the last level is the biosphere, which is our Earth. Okay, and cells can separate into 2 kind of organisms, which normally we can the general one is the multicellular organisms, which is the organism that is made up from a bunch of cells. Like people are the multicellular organisms, because when it was the multicellular organism, it means that like we have many cells, right? And when many cells are formed, they form tissues. And then many tissues are formed, they form organs, and they form a system, and system form organism, which is the living things, like the individual living things, like tigers, snakes, people, like that. And then the next one is the unicellular organism, which is it's hard to find, because it is the one single cells. So when it is just one single cells, that means they can't even form a tissues. So they're gonna stop the organizations, like the level organizations, just in the cells, like that. Next topic is the natures of science. So science objective is to understand how the nature works through observations and reasonings, because in science we can just predict it and say that it's true, right? We need to prove it and give the reasons. So there's two type of reasons that science uses. One is the deductive reasons, or in Thai we call itแบบนิรนัย. So it's the general principle to make predictions that is usually true. Like if we drop the ball will fall down, like that. And the second one is the inductive reasoning, which is the specific observations to develop general conclusions. Well, this one, it's a bit like complicated. So when we finding for like in inductive reasoning, scientists are usually using this method because like to gain more understanding, scientists use the method that is called the scientific method. So how to use scientific method? First, we're gonna know, after like we know our topic already, first we're gonna observe first the observations. So we're gonna observe it, that like how is it going, what is the problem, like that. And then second is the hypothesis formation, which is like when we need to think that like what could go wrong in this problem, like what's make this problem. And then when we think about it already, third is the predictions. We're gonna predict about how we're gonna fix the problem and how it'll come out. Like from the hypothesis, which is when you think that, for example, you think that because of A, that makes this problems go wrong. Then for predictions, they're gonna predict that like that we need to fix A to, and then what will come out is that the problems will gone, like that. So when we predict it already, we're gonna start doing it, which is the experimentations. Well, in the experiment, we have two things, which is the test experiment and the control experiment. Which the test experiment is the real test, and the control experiment is just like the things that used to help the experiment. For example, we're gonna do experiment about vaccines, like that, about vaccines, and the test experiment is the group that, for example, in the experiment we separate people into two groups, right? Group A and Group B, and then the Group A is the test experiment, which is the real test. So Group A will be the group that we're gonna give the real vaccine, and then Group B is the control experiment, which we're gonna give something else, which is not vaccine, like for example, like vitamin, like that. So we're gonna see. Okay, so this is Group A and this is Group B. We give vaccine to them, and for this one we give something else that is not vaccine. This one is vaccine. And then why do we need to have Group B, right? This is because that when we have Group B, we can compare easily that like what like the results, like that. If we see is that like the result of Group A and Group B is like very different, so we can conclude, which is the last step, is the conclusions. After we see the experiment, so we can conclude that like, is it work? Is our hypothesis real? Is our predictions real? Like that. And then, yes, this one is the philosophy approach to science. So the reductionism is to break down the process. For example, like if the process are like many things, what we can do is we can just like break down it and look at it just like one part of it. For example, I want to look at this part. So when I just focus about this part, it will be easier to understand. And for the system biology is when to forecast about other part, because in some process we can't, or like in some things we can't break it down, or maybe some things we just like, when we forecast it on all part, we're gonna have the new informations about it, like that. Next is the Darwin and evolutions. So Charles Darwin's are famous about the evolutions. He used around 30 years to observe and study before he published out the on the origins of species by means of natural selection, which notes that he isn't the first one to propose about this, but he just the first one who published it out. So Charles Darwin, he sees that the characteristic of the similar species develop from place to place. Some species have similar characteristics. Then, after he sees like all of these things, he made an important associations. That's individuals with features that give them advantage in their environment are more likely to survive. Does this mean? This means that, like, for example, the rabbit, for example, uh, usually rabbits have strong legs, right, to like jump like that. But if from this one that like the individual with the features that give them advantage like that, for example, there's this one rabbit, the rabbit named A, right? The A rabbit, he can jump more than others rabbit. So that's mean that like his legs are stronger than other rabbits, and this give them advantage. These features, which is the stronger legs, give them advantage in the environment, which is like, for example, they need to run away from the snakes like that. This will help them because they can jump faster than others, and then they will have more chance to survive. And if these advantage pass on their offspring, then this considers as the evolutions. So this is very important, because if the same rabbit, if the rabbit A, he can't, like when he have a baby and he can't pass on the strong legs to the baby, that wouldn't count as the evolutions. But then if he pass on this features on his offspring, which is his baby, which is a stronger legs, this is the evolutions. So the rabbit A baby have the stronger legs, and then the baby of the rabbit A have a baby. The baby of the rabbit A baby will have the stronger legs. So it will be like continue like that. So that is the evolutions. And then the populations will change over time. Therefore, the Darwin called this as the natural selections. Okay, so there's also the evidence that support Darwin's, which is like the fossil records, the first one. Fossil records, like because like the fossils make us see that the past from the same one, the rabbit A again. So if we in present we found the fossils about the rabbit fossils, and we can see that like the rabbit fossil, they have like a very thin leg like that. So we can see that the past rabbit and the present rabbits are very different. The past rabbit maybe can't survive easily because they have a thin leg and they can't jump fast like that. So this is the evidence. Next one is our Earth's age, which is 4.5 billion years old. Next is the mechanism for heredity. And at that time, Darwin, he can't explain about this because he didn't have enough like information about it, but he knows about it. Next one is the comparative anatomy. So the animals that have the same origins will have the similar anatomy, like the cats and the dog. If you take them on X-ray, you can see that their bones are very similar. That is because of the origin. The origins are the same. The last one is the Molecular evidence. What is the molecular evidence? It is the genetic code of the same origins though similar. Okay, the next topic is unifying themes in biology. Okay, so the cell theory is that all organisms are composed of cells. The molecular basis of inheritance are those things that pass on genes. For instance, like genes itself, DNA, sequence of nucleotides, like that. Let's go to the next chapters. The chapters 2. So it is about the nature of atom. So all matters have mass and is composed of atom. Okay, so why? Sorry. So it's composed of atom. Every atom composed of three things, which is the proton, neutron, and the electron. Okay, so proton is the positive charged particles, which we can find it in the nucleus. Next one is the neutron. Neutron is the neutral particle, which means they're not positive and they are not negative. Oh yeah, that's right. They're not positive and they're not negative. And we can find it in the nucleus. The last one is the electrons, which is the negative charged particles. We can find it surrounded area of nucleus. So it's the only one that is not in the nucleus. So it's atom. This is the nucleus, and this is the proton, and then neutron, and this is the electron, like that. Okay, so atoms occurs naturally 90%, and atoms because of like there's also some atoms which is like some element which made from the lab by human, which is man-made, like that. And elements are made up by one type of atoms and have only one atom also. Next is the atomic mass, meaning the amount of substance so atomic mass is the amount of substance, but in this case, it means just to sum of the protons and the neutrons, because of the electrons they are very, very, very bow. What's it? They're very lightweight. Yeah, they're very lightweight. So we didn't consider it in the atomic mass because like it won't change anything at all. And the unit of the atomic mass is the 1 Dalton or atomic mass unit. Next is the isotopes. Isotope is the atom with the same element but has a different amount of neutrons. For examples, what should it be? Chlorine, for example, they have like 12 like that, and this one have 14 like that. So they're isotopes. Next is the atomic models. There are many atomic models, but in bio, we're gonna take up just two atomic models, which is the Bohr atomic models. So Bohr believed that the electrons are surround the nucleus in the same way that the solar system works. So what he means that like this is the atoms. No, sorry, this is the nucleus. He think that like the nucleus work like a sun, like that. Then this is the shell of the electron, and this is the electrons. Like see, it's look like the solar system. He think like this. He think this is how the atom looks, like that. But then the next model is the quantum atomic models. So these models come out against that the electrons doesn't look exactly the same with the solar system. They didn't orbit the nucleus in the obviously path. So what they mean is that he means that like the electrons doesn't leave like very obviously like that, but like they look like the electron clouds instead in this atomic models. So what he means is like this is the nucleus, this is the atom, and then the electrons are like, it is like the cloud, like the glumor. In Thai we call it the glumor, like that. So we cannot know exactly how electrons are, like how electrons form. We just know that like they're surrounding the nucleus like that. Next is ions. Ion is like the unbalance of protons and electrons. Treat the atom with the charge, either positive or negative. This is called as ions. For the cation, it is the positive charge, the atom with the positive charge, and the anion is the atom with the negative charge. Next is the octet rules. Octet rules is the rules about that valence electrons, they want to be 8. Like, because not all the valence electrons have 8 electrons, right? So this rule is about that, like the valence electrons, they want to have 8 electrons in their shells to make themselves stable. They believe that have 8 electrons make the atom stable. That's why. And this means that the inner gas group is the non-reactive elements, since they are already stable, because the inner the inner gas group is already 8. They already have 8 electrons in the valence electrons. So chemical bonds. Chemical bond is the process to make the atom stable. So, because as the octet rules that electrons want to have 8, the atom wants to have 8 electrons. So they're gonna do bonding to make the process as a process to make themselves 8, like that. So we have 2 kind of bonding, which is the ionic bonds and the covalent bonds. Ionic bond is to form by attractions of the opposite charged particles, while the covalent bond is the when the atoms share the valence electrons. So here, unlike the ionic, that like they give it. So this is the ionic. For example, this one, you can see that like the right one, this one, they already have 7 electrons and they want to have 8 electrons. So what they do is they steal these electrons from the another atom for themselves. They steal it, so they have 8 electrons, like that. But for the covalent bond, what they do is they sharing the electrons. They didn't steal it. Here, so we have 3 types, which is the single, double, and the triple. Single is when we use just two electrons, but when like they come out here, they will come out as one line, which is one. So that's why we call it the single covalent bond, because like we have just like one line, because 2 electrons form one bond like that. That's why. Double covalent bond, we use 4 electrons, which is 2 bonds. And triple covalent bond, we use 6 electrons, which is 3 bonds, like that. And group of atoms. So molecules is the group of atoms that have only one type of element, while the compound is the group of atoms that contain more than one type of elements. Carbons and organic compounds. Organic compound is the compound that contains carbon, because if they didn't contain carbon, they will call as the inorganic compounds. And one carbons can form up 4 single covalent bonds, and these carbons are the backbones from the basis of organic molecules. Next is the electronegativity, or we call it En. This one is the differences in the electronegativity shows how electrons are distributed in the covalent bond. So the nonpolar covalent bond, that's mean they equally share the electrons, but the polar covalent bond, that's mean they are unequally sharing the electrons, and that's mean they're gonna create the particle charge like that. Next one is the water. 2 of 3 of organisms' bodies are water. The most outstanding chemical properties of water is the ability to form hydrogen bonds. This is like the most unique one, like the most unique properties of water. So, the hydrogen bonds is the weak bond between the two water molecules. Therefore, it's easily broken by heat, because like the hydrogen bonds, they didn't as strong as those ionic bond and covalent bond. They are easily broken. That's why. And the bonds between oxygens and hydrogens are highly polar. Highly polar, you need to remember it. It's highly polar, where the oxygen is negative and the hydrogen is the positive charge. Next is the physical property of water, which is the cohesions, which co mean together and hesions mean glue, which is stick. So that's why cohesion is when the water molecules stick to other water molecules by hydrogen bonding. While another one is the adhesions, which is the water molecules stick to other polar molecules by hydrogen bonding. So the difference from this one and this one is just only that cohesion is stick to other water molecules, and the adhesion is the water stick to other polar molecules. That is just the only difference. Okay. And the acid and bases. So pure water is the middle. You can see from this one. See that the pure water is the middle one. They're not bases, and they're not base, and they're not acid. Let's know about acid first. So acid is any substance dissociates in water. So the stronger the acid, the more ions hydrogen it is, and the more hydrogen it produces, and the lower pH it is. Bases is any substance that combines with water. They didn't like associate from the water like that. Okay, so these are all from the chapter 2. So let's go to the next one, the chapter 3. Okay, so we're gonna start from the carbon. So all organic compound consists C bond, or the carbon bond to other elements. So example, one of it is the hydrocarbons, which is the molecules that contain carbons and the hydrogens. And mostly the hydrocarbons are non-polar, like those gases or oils, like that. But there's some cases that it can be polar, which is when the hydrocarbons has the functional group inside them. The functional group is like the groups that added the chemical properties where the biological molecules made from. Okay, so it's these guys are the functional group. So not all of the structures are the functional group. The green highlights, that was the functional group. You see that? Like that. So here, CH, right? CH. So this is the hydrocarbon. So that's mean hydrocarbon with the functional groups. So that's why they're polar, because the functional group is polar. Okay. So let's know about this first, by the way. So we have the hydroxyls, which is OH, and we have the carbonyl, which is the O double bond C. I'm sorry, O double bond C. And for carbonyl, we have two types of carbonyl, which is the ketone and the aldehyde. Ketone is like the carbonyl that has H, which is hydrogens, but the aldehyde is, oh sorry, I'm so sorry. Ketone is the one that doesn't have hydrogens, while the aldehyde is the one that have hydrogens. Next is the carboxyls. Carboxyl is I think it's the mix of the hydroxyls and carboxyls, because you see this. Hydroxyl is OH and carboxyl is OC, right? And then the carboxyl is the the O double bond C with the OH. See, so they have both carbonyl and the hydroxyls. That's why. Next one is the amino. So it's this one, and H2, and then the sulfhydryls, which is the SH. S come from the what's it called? Sulfur, yeah, the sulfur. Sulfur. Yeah, and then the H from hydrogens, the same one as always. And the next one is the phosphate. Phosphate they use so much in this. Yeah, phosphate used so much in this topic, so we have to remember it. And then the last one is the methyl. Yep. So next, we would like to know about the isomers. So isomer is the molecules with the same molecular. So we have two types of isomer. The first one is the structural isomer. Here. The structural isomer is the difference in structures of carbon skeletons. They switch place in the skeletons. On the other hand, the stereoisomer is the one that have like the same carbon skeletons, but they just like in the different arrangement. The different arrangement means just like they just like switch the side, like from left to right like that. And we call that one the chiral. That's mean like to spin left or to spin right. Do I have I don't think I have. Yeah, here. This one is the example. Here, the example. We see that the structural isomer is when like they change in the skeletons. See, like this one is the base one, right? And the structural isomer you can see here that they just change the place. While the stereoisomers they didn't change the place. They just like switch the side, like the mirror. Just switch OH to left here, and they switch hash to right like that. Okay, so next we're gonna get to the macromolecules. So the macromolecules are made of polymers, like many polymers, and many polymers are made from the monomers again. So monomer is the like small things that like a bunch of them can join and create into the polymers. And then the polymers build the many monomers, like I said, like that. Let's know the type of macromolecules. So, first one is the proteins. So proteins, the monomers of the proteins is the amino acid. This means that like many amino acids forms the polymers and many polymers form proteins again. Like that. And then the second one is the carbohydrates. Carbohydrates monomers is the monosaccharide. Third one is the nucleic acid. The nucleic acid monomer is the nucleotide. And the last one is the lipids. Lipids, they aren't made from the monomers, but they are considered as the macromolecules. They're made up from those like fatty acid. And then how to make the, and I mean like the process to make the macromolecules. The process is we have tools, is to build and to break, right? To build, we need to use this one, dehydration synthesis. So dehydration synthesis, it's same as the name. So D means like to take out. Hydration is like water, something like that, right? So dehydration synthesis is to remove out the water to join the monomers in, like that. And then the next one is the breaking macromolecules. It is the opposite with the building. Building is to remove water, right? The breaking we uses the hydrolysis, which is to add water, not to remove. We're gonna add water into the break. Oh, yeah, we're gonna add water to break down the monomers. I'm sorry, there's no two. There's this two. I'm sorry. So to add water in to break down monomers. I should put a comma here. Okay, so let's start with the first macromolecules. You see, right? Carbohydrates is the macromolecules. Okay, let's start from the first one. Carbohydrates. Carbohydrates, they have the ratio, right? The molecular ratios of the carbons, hydrogens, and the oxygens is 1 to 2 to 1. That is the ratios. And these three are the composed of the carbohydrates. Okay, so this is the formula of the carbohydrates because like it's like this, right? And when if we put 4, it's gonna be like this. Distribute 4 inside. 4 times 2 is 8, and see, 4. See that like the ratio is 1 to 1, like that. So carbohydrates has a lot of CH covalent bond, and the CH covalent bond, they hold a lot of energy. So that's mean the carbohydrates is the good energy storage molecules. That's why like when we eat carbohydrates we have power. That's why. Okay, so there's 3 major groups of the carbohydrates. First is the monosaccharides, which is the monomers, as I said. The second one is the disaccharide, which is the polymer with 2 monomers already. Third is the polysaccharide, which is monomers is with the polymers with many monomers. Let's start from the monosaccharide first. Monosaccharide is the simplest carbohydrate because like there's just the monomers. So examples of the monosaccharides are the glucose, the fructose. So glucose is the, what can I say, like the original one, the base one, and then fructose is the isomers, the structural isomers of glucose. And then the galactose is also isomers, but it's the stereoisomers of glucose. See this one. See, this one is like the base one, and then this one is the isomer, and this one is the isomer. Okay, and yeah, glucose, they can become a linear, which is like normally, and they can become a ring too because this side they don't like water, and this side they like water. So when like there is a water, this side they will like roll inside like this. See, they will roll inside and become a circle. And when they roll inside, they can become 2 forms, which is the here the OH, the hydroxyls. They are if they are here like in the down spaces, they're gonna be the alpha glucose. But if they are on top of the hydrogens, which is the opposite side of this one, they're gonna be the beta glucose, like that. And the next one is the disaccharide. Disaccharide is the 2 monosaccharides or the monomers linked together by dehydration synthesis. For examples, sucrose, lactose, and maltose. Lactose, we know this one, right? It is the one that we have in the milk, right? And is used for the sugar transport and energy storage also. Last one is the polysaccharide. Polysaccharide is the long chain of the monosaccharide. There's 4 major classes. One is the starch. Starch is the energy storage in plant. They only have in plant. Next one is the glycogen. The glycogen is the energy storage in animals. So there's the opposite, right? And then the cellular. Cellular is the molecules of plants in those like fibers or like plant cell wall. And then the last one is the chitin, which is they give the structural support in like insects or like the arthropods. I think that's what they call arthropods. Next one is the nucleic acid. So nucleic acid, the monomers are the nucleotide. So they come like what nucleotide contain. They contain three things, which is the sugar, the phosphate, and then the nitrogenous bases. So sugar, we're gonna have two types, which is the deoxyribose and the ribose. I think if it deoxy, that means it is the DNA, but if it ribose, that means it is the RNA. Okay, next is about let's know a bit information about the nitrogenous base first. So we're gonna have 5 type of nitrogenous base. It is the A, G, C. This is G. Sorry, let me write again. G, C, T, and U. So A and G's are the purines, which is like there are 2 of them. While the next one is the pyrimidines. Pyrimidines are just like one of them, right? So pyrimidines are the CTU. CTU is like the cytosine, the thymines, and the uracil. The purines, I forget to tell you, A is the adenine and G is the guanine. And like in both DNA and in RNA, we having A, G, and C. But if it the DNA, we're gonna have T include. But if it the RNA, instead of T, we're gonna have U include. All right. Next one is the deoxy. Oh yeah, so it's the DNA, right? The deoxyribonucleic acid. So DNA, we are composed of genes, right? Encode, which is the encodes information for amino acid sequence of proteins. See the sequence of bases? We have 4 of them, which is A, T, G, G and C. And the formula of it is the double helix, which means they have like 2 lines, and the 2 lines like they twist around each other. And the the bases, the nitrogenous bases, they have like the pairing rules, which A need to pair with T and C need to pair with G. See, you see here, A with T, C with G, and A with T. Like A doesn't have to be on the left side. They can be in both sides, but they need to be with a T. T and A, G and C, like that. Okay, and here we might don't know that like where is the nucleotide stand. So we can look in the the sugar phosphate bonds, which is this one, this line that like they're twist around each other. Which is this one. is the backbone. We call it as the sugar phosphate backbone because here you see like the little p, the yellow p. This is the phosphate group, and then this one is the sugar. See, so the phosphate will bond with the sugar, and how will it bond? The things that bond between them is this one, the phosphodiester's bond. That's it. And here also the N base, it's around here at the moon. Alright, and then another things that like pairing, not just AT and CG, is this one. See the 5N here, the 5N, you can see that the last one is P, and then the 3N, the last one is the, what's it called? I forgot. I'm so sorry. It's the sugar. Sorry, the sugar. So that's why. And it's also pairing because like if one side is 5N, another side has to be the 3N. It can be like 5 and 5, and it can be 3 and 3, like that. Okay, next one is the ribonucleic acid, which is the RNA. It is similar to the DNA except that they contain ribose instead of the deoxyribose, and they contain uracil instead of thymines, and they are single strand. They're not the double helix. They're not like this. They're just like this one, like the single strand. This one, like that. Okay. Here, this is the RNA. We see, right, that they're just like one line, like that. Okay. Oh, and ATP is it is the primary energy of all cells. Like it's the primary energy. Oh, the full name of ATP is the adenosine triphosphate. That is the full name of ATP. Next is the protein. Okay, so protein functions. They have many functions. They can it is the enzyme catalysis. 2 is to defense. 3 is to transport. 4 is to support. 5 is motions. And 6 is the regulations, and 7 is storage. So there are 20 mono acids. I'm so sorry. There are 20 amino acids in one chain of proteins, and in each chain is a polypeptide, and the amino acid linked together by the peptide bond. Okay, so the amino acid structures, they also have structures. The structures are composed by 5 things, which is the central one, the carbon atom, and then the amino group, and then the carboxyl group, and then the single hydrogens and the R group. The R group is the things that make amino acid structures unique. And the amino acid structures, they have 3 types, which is the nonpolar, polar uncharged, and charged. Okay, so see here, nonpolar, polar uncharged, and charged, right? Here. You see that like they have charge here. See, negative, positive, like that. Here, positive. Yep, that's why. Oh, and this one it's the polar, right? This is the polar. That's why it's the polar, but they're uncharged. Okay, and the protein structures, we have all of it is 4 levels. In total, 4 levels, sorry. So the first one is the primary. Primary is just the sequence of the amino acid. So like the many amino acids is just drawn together like that. Amino acid 1, amino acid 2, like that, drawn together like that, as a long chain. The secondary structure is, oh, I forget to write the primary structures, I'm sorry. The secondary structure is to interact a group of in the peptide backbones, the hash bonds between amino acid, and they can got twist in two ways, which is the alpha helix and the beta sheets. This one is the alpha helix. See its twist here. This is twist. Like it for me, I think it's look kind of like the DNA because like they are twist, but they have only one chain, not two chain. And then the beta pleat, see it's like การจับจีบ in Thai because like see that like there are fold here, fold and fold like that. It's like a sheet that can fold like that. The third one is the ter tertiary structures, which is the final fold shape of the globular protein. Globular proteins mean like the circular, like the circular shape, like that circle shape. So like it's mean that like many of beta pleat and the alpha helix, many of them are formed together. They are formed into the circle shape like that. And then the last one is the quaternary structures, which is the arrangement of individual chains formed together. So the quaternary is like this one being together a lot like that. And the chaperone protein is to help the protein to fold correctly, because like see, this is the process. This is the chaperone proteins, and then there's a misfold proteins that come in here, and then there are the APT, which is the energy that help it. See, it starts to shape normally, and then here they shape normally, and then alright, they are fully processed into the correctly folded protein. Then there are the denaturations, which is when the protein lost the structures and the function. This happened by many factors like temperatures or like the pH. I think pH also. Temperature, like for example, you grill the salmon. The salmons they're gonna lose. The salmons, they have proteins like or like you grill the porks like that. The porks, they have proteins also. So the proteins in the pork, they're gonna lose the structures and the functions because they got grilled in the hot temperatures. The last macromolecules is the lipids. The lipids is the last one. So lipids is the group of nonpolar compound. They are hydrophobic and they are insoluble in water. That's this one. That's why humans, we are not like we not get mixed in the water when we shower, and they are not polymers because they have no monomers, but they are the macromolecules, and they are the excellent energy storage. So diglyceride composed of one triglycerols and three fatty acids. So here you see the red atom. Yeah. So this one is the glycerols, and this chain is the fatty acid. One fatty acid, two fatty acid, and three fatty acid, like that. This one is like the tail of the lipids. Not lipids. Yeah, lipids, I think. No, it's the tail of the phospholipids. Here, this one. And okay, fatty acid, we can have two types, which is the saturated and the unsaturated. So saturated have no double bond between the carbon atoms, and the saturated are the animals' origins. They have high melting points, and they are solids in room temperatures. For examples, like the butter like that. You can no, I think yeah, the butter. I think it's not. I'm not sure. I'm sorry. And then the second one is the unsaturated, which has like one or more double bonds. They are low. They have low melting point, and they are the plant origins. They are liquid in the room temperature. We can see like from like those oils, like the vegetables oils. They are. You can see that they are solid, right? When you use it to cook, right? Like that. And the phospholipids is the composed of the phosphate group, which is the polar head, the head of the phospholipids, and then the triglyceride as the tail. So this one is the phosphate group. No, this is the choline, I think. Choline. And this one is the phosphate group, and then this one, what I have showed before here. See? So this is the glycerols, and here is the fatty acid chain. See? If they have double bond, that means they are unsaturated, right? And this one, we don't have the double bond, which means they are the saturated one. Okay. Oh, and the head are polars. Here are polar, and the tail are non-polar. Okay, and we have the micellus, which is when the lipid molecules are entered with the polar head, which is the hydrophilics. That means love with the water. They arrange with the polar head toward the water, and the non-polar tail, which is the hydrophobic, away from the water, and it's gonna form like this. Here you can see that like the tail, they don't like the water, so they get inside, while the head, which love water, they get like outside, like that. So this one, we can hide it from the micellar water, which we use it to remove our makeup, because that our makeup basis is the oil basis, and the micellar water's they gonna like stick. How can I say, like they're gonna stick our makeup out with the micellar water, like that, like stick our makeup from our face out, like that. And then the phospholipid bilayers. This is the more complicated structures, where the two layers formed by the polar head point outward and the non-polar tail point inward, like this. So here, so they point like this. Yep. And then, oh yeah, this. See what I have right in the slide, that like it's protect the water to not get in, because the tail, they don't like the water, right? So even the head, they like the waters, but when the water try to get in, right, the tail's gonna like push the water out, because they hate water like that. That's why. And another structures is the steroid, which is the ring structures. Steroid, we can see like they mostly come in like come with the hormones, I think. Here, like the cortisol hormones, the testosterone, which is the male characteristic hormones, and we have the vitamin D too. Also, as the steroid. So our bodies can produce steroids. That's why like one reason that the doctors, they don't allow us to use steroid for too long, because like if we use steroid for too long, our body, they will stop producing steroid, and we won't have steroids anymore, and we have to use steroid instead, like every day, like that. Okay, next one is the chapter 4. Okay, so the chapter 4 is about the cell, right? So we're gonna first start from the cell theory. So Robert Hooke is the one who discovered cell in 1665, and then Anton van I'm not sure how to pronounce his name. Sorry if I didn't pronounce his name correctly. Is it Leeuwenhoek, I think? So he observed the living things and he called them the animalcules. Animalcules, which means the little animals. Then I also not sure how to pronounce their names, but these two people they proposed about the cell theory. So let's know about the theories. The theories is about the theory says that all organisms are composed of cells, and cells are the smallest living things. The last one is the cells can happen only from the pre-existing cells. That means that cell they can't just like made up themselves by themselves like that. And about cells is that cell size is limits and rate of diffusion, which is การแพร่ in Thai. They are affected by four things, which is the surface area, the temperature, the concentrations gradient, and the distance. Okay, and the surface area to volume ratio is 100 to 1000. Next is the microscope. So most cells are less than 50 micrometers and in diameters, and human eye resolutions are just about like 100 micrometers. So that's why we need to have the microscope to see the cells, like obviously like that. So there's a two type of microscope, which is the light microscope, using the magnifying lens with the visible lights. This one will resolve the structures that are 20 nanometer apart. And next one is the electron microscope. This one will use the beam of electrons, and they're gonna resolve structures that are 0.2 nanometers apart. So they can separate into two types, which is the transmissions electrons microscope and the scanning electron microscope. So the transmission is to transmit the electrons through the materials. And then the scanning is to beam the electrons on to the specimen. I'm sorry, I forget T. Specimen's surface. And then the next one is the type of cells. So we're gonna have two types of cells. The first one is the prokaryotes type. The prokaryotes, they are about 1 to 10 micrometers. It is the simplest organisms, and they didn't have the nucleus. So they have the DNA in the nucleoid, and see, they don't have the nucleus. And the cell walls are outside the plasma membrane. They also have the ribosomes, but yeah, they also have the ribosome, which is the protein synthesis. And the only two prokaryotes are the archaea and bacteria. So in our world, we only discovered two types of prokaryotes. Maybe there are gonna be more in the futures, but in the present, there are only two types, which is the archaea and bacteria. So this is how they look. See, they have the plasma membrane, they have the cell wall, and they have the capsule. They have I think it looks like the pills also. And they have cytoplasms, same as the eukaryotes, and they have the ribosomes and DNA, same as the eukaryotes. Okay, so forgot to go deep in the information. So bacterial cell walls So these two are the organelles of the prokaryotes. So the organelle one is the bacterial cell walls. They are the strong cell walls, and the function is to protect and to maintain cell shape, and it's helped to prevent too much water, help to prevent too much water uptake also. And then the flagella, which is like the tail, see, like this one, the tail of the prokaryotes. They only have in some prokaryotes, like not all prokaryotes gonna have them, and they're used for movement. Yeah, so like having the flagella helps the cell to move better, like that. And their proteins, and they can have the in one prokaryote, the flagella can have from 0 up to 1 up to 2 or more. And then the next type, which is the eukaryotics. Eukaryotics is they are about 10 to 100 micrometers. They have membrane-bound nucleus. They are more complex than the prokaryotes, and the hallmarks, or like the specialties about them, is the compartmentalizations, or we call it 太干板素. And they also have the cytoskeletons, which help to support and help to maintain the cell structures. Here, this one, you see, like these things, they help to maintain, help to maintain to the cell structures, the structures of the cells, like that. So we have two types of the eukaryotics, which is the plant cell and the animal cells. Have some difference, and some are same in these two types. So the first organelle is the nucleus. The nucleus is the storage of the cell's genetic informations. In the nucleus, we have two things, which is the nucleolus, where the ribosomes RNA synthesis take place, and the nuclear envelope, which has two phospholipids bilayers, which I have mentioned in the chapter 3, and the nuclear pore, which controls the passage in and out, which is this one. See, they contains like what to get in and what to go out, like that. See, this is the nucleolus, where the ribosomes take place, and yep. Oh, and this is the envelope, the nuclear envelope. And DNA divide into multiple linear chromosomes, and when the chromosomes like mix together with the proteins, it's called as the chromatins. Next one is the ribosomes. See that, right? That here, ribosomes. It's also take place in the nucleus, right? So ribosomes, they take place in many places. One of them is the nucleus, and another one is in the ER. So ribosomes their duty is like to make it is the cell's protein synthesis machinery, which is like to produce proteins, like that. They can be in both prokaryotes and eukaryotes. They're just like one of the few organelles that in both prokaryotes and eukaryotes, and composed of ribosome RNA and protein complex. And they can locate freely in the cytoplasms or stick with the internal membrane. So what is the meaning of this one is like in the cells, right? Oh, let's go to this place. In the cells, we can see that there's the jelly-like form, which is the cytoplasms, that one. So the ribosomes can just like live freely on the cytoplasms, like that, like without sticking to any other organelles, like that. And they can also stick on the other organelles also, which is like the nucleus. See, these orange spots are the ribosomes, and also this one, see, on the ER also. The rough ER contains ribosomes also. Okay, let's go back to the endomembrane systems. Is the series of the internal membrane, and the members of the systems, we have three members altogether, which is the endoplasmic reticulum, which is the ER, and the Golgi apparatus, which is the another membrane, and also the lysosomes. And what does this system do? The endomembrane system, it's help to allows the cell material to flow through, and they are where the proteins and lipids are made and modified and transport. Okay, because each members have their own duties in making, in modify, and in transport, making lipids and the proteins. Okay, so the first member is the endoplasmic reticulum, the ER. We also have two types of ER, which is the rough ER. It is rough because of the ribosome make it rough, and there's the synthesis of the proteins. Then it's sent to the lysosomes or the plasma membrane, send the proteins to them. The next one is the smooth ER. So smooth ER have very few of ribosomes that make it smoother than the rough ER. And for the smooth ER, they make lipids instead of the proteins. And they have many functions. I mean, they can store the Ca, I think that it's the calcium, and they also detoxifications also. The next member is the Golgi apparatus. It's the flattened stack, which is these pictures, and their duty is to packaging and distribute the molecules. And they have two phases, which is the cis and the trans. So this is cis and this is trans. Cis is the ฝั่งที่นูน and trans is the ฝั่งที่เว้า. And they have vesicle transport molecules to destination. So the Golgi, when they pack the things already, they're gonna send the vesicle, for example, this one they can see they are about to make it. When they finish make the vesicle, they're gonna send it out and transport it to the other places like that, to the destinations, I mean. Okay, and the lysosomes only have in animals is the membrane bonds, digestive vesicles, and the lysosomes are made from the Golgi apparatus. And there are two enzymes in the ribosome, which is to break down the macromolecules, and another one is to destroy the matter taken in by phagocytosis, which is การแยกอาหารของเซลล์. So here, this is the Golgi, right? And then they're gonna send out the lysosomes. See, the lysosomes are making, and they send it out here. And then the lysosomes, when they found the vesicle, they're gonna form, and they're gonna digest the food for the cells. I mean, yeah, they're gonna digest food to give the energy for the cells. To give the food for the cells, I mean. Okay, and the next organelles is the vacuoles. Vacuoles only have in the plant. The vacuoles structures in plants. See, the structure is in plant. They have many functions. It depends. So there's have three types of vacuoles. First one is the central vacuoles. They are in the plant cells, and it helps to maintain the pressures. Second one is the contractile vacuoles, which is in some fungi and the process. And then the last one is the storage vacuoles. I don't have the pictures, I think. Oh, this one here. Do you see this big thing? This is the vacuole. This one is the central vacuoles. Okay, the next one is the mitochondria. Mitochondria are found in all eukaryotic cells. It's used for the cell respirations, which is the energy. They are bound by two membranes, which is the outer membrane, and between the outer membrane and the inner membrane, which has the cristae, they're gonna have the things called the intermembrane space, which is like in between the outer membrane and the inner membrane. This is the outer, this is the inner membrane. See? And then inside the inner membrane, it's the matrix. See? Matrix. All of it are the matrix. And the inner membranes has proteins that carry out the oxidative metabolisms. And mitochondria are only one of the two organelles that have their own DNA, not just like the DNA in the nucleus, which is the DNA for all cells. This is their own DNA. Next one is the chloroplast. Chloroplasts only have in plants, and they contain the chlorophyll for the photosynthesis. See, that's why they're green, and that's why the plants are green, because they do the photosynthesis. And in plant cells, they are in plant cells and some eukaryotes. They are surrounded by two membrane, which is the thylakotes, which is the membrane sacs inside inner membrane, and the gyna, which is the sacs of the thylakotes. See, this is the gyna. Oh nope, I think this one. See it like that. And the stroma. Stroma is the fluid matrix inside the chloroplast. See? So inside, you see this one is the stroma. Stroma is the fluid things inside. And see, chloroplast is another one of the only two organelles that have their own DNA. So the only two organelles that have their own DNA is the mitochondria and the chloroplast. They have reasons, but I'm gonna explain it later. Let's go to the next one. Is the endosymbiosis. Okay, so when the prokaryote starts to living in then another eukaryotes, and it's become its organelles, and the two cells it is the symbiosis, which is like ภาวะการพึ่งพากัน in Thai. See, so this is the eukaryotes. Then there are some like unknown prokaryotes come and like live inside the eukaryotes like that. And this one, like sometimes they live until like they become the organelles like that. That's why the mitochondria and the chloroplast have their own DNA, because this is the origins of the mitochondria and the chloroplast. That's mean both of them are the unknown things, the unknown cells that becomes the organelles in the eukaryotes. See, this is the origins. Okay, the next one is the cytoskeletons. Cytoskeleton is the network of the protein fibers, which they support the shape of the cells, and they keep organelles in the fixed locations. There are three types of fibers. One is the macrofilaments, or we call it as the actin filaments. This one there are two protein chains twist together, like this pictures. See, they're twist together, like loosely twist. And then the next one is the macrotubules. It's how the movement of the cells and their organelles, and it's the largest fibers in all three. Just these pictures. And then next one, the last one, is the intermediate filament. The size of this one is between the actin filament and the microtubulus, and it's very stable. It's usually not break down. For example, like the keratin, which is in our hair, like that. The last one is the centrosomes, which is only in the animal cells. The centrosomes they have the things called the microtubules, which is named same as this one. See? It's this one. It's the same things. So microtubules is the organizing center. This one, you see it? Okay, so the centrosome meaning is the pair of centroles. So centroles is two centroles is one centrosome. Centrosomes, I mean, and organized modules for the cell division. This one. And for the cell movement, the actin filaments and the microtubules or both they are going to help about the movement of the cells. And also the flagella and the cilia, they have 2+9 arrangement of the microtubules. And the flagellae for this one, they are not same as the prokaryotes flagella. And the cilia are shorter and more numerous in this one. Next one is the eukaryotic cell walls, which is all in the plants. This one is different from the prokaryote cell walls. The prokaryote cell walls and the eukaryote cell walls have different. It is different. So it is the cell wall. It is in plant cell, in fungi, and in protists. In plants and in protists, it's called as the cellulose. The cell walls are called as the cellulose. And in fungi, it's going to be called as the chitin. And the plants, for the plant cell wall, they're going to have both primary and a secondary cell wall. Like this pictures. This is, you see this? This is the darker green, the dark green, that was the primary wall, this one, and the inner one is the secondary wall, like that. And because of that, the animal cell doesn't have the cell walls like the plant cells. That's why we have the things that call the extracellular matrix, ECM. They form to protect the cell, the animal cells. So they form as the protective layers over the cell surface, and they are rich in collagens, and then the integrins links ECMs to the integrins, which is this one. They links the ECM to the cytoskeletons. So they link down like that. See that we have like many collagens here. That's why like our faces have collagens, because we are animals. And cell-to-cell interactions. So the surface proteins give the cell's identity, and to make contracts, cells are reading each other and react. And the glycolipids, it is the tissue-specific cell surface markers. And the MHC protein, it's recognized of itself and the non-self by the immune systems. So the immune system helps them. So connections. So connections, we have three types based on the functions. First one is the tight junctions. It's connect the plasma membrane in the sheets like this. See, in the sheets. Next one is the anchoring junctions, or we call it as the desmosomes. It's attached the cytoskeletons of the neighboring cells, like this. And the last one is the communicating junctions, or the gap junctions. The chemicals or electrical signal pass from one cell to another adjacent cells. Just this one. Okay. Another one is the plasmodesmata, which is only in the plant cell. So it's specialized open in the cell wall. See, they are already open, and the cytoplasms of them are connected. Like the two cytoplasms are connected, as you see, and they are similar to the gap junctions also. This is all of my summarized note presentations. Thank you for listening and for your attention.